Good day, everyone. I am Gabriel or Cabrera, a student from Computer Engineering Department, first year in Colegio de Muntinlupa. So, today I will present a presentation about development of a robotic system for automatic organic chemistry synthesis. Why do we need to study robotics? Robotics technology influences every aspect of work and home. Robotics has the potential to positively transform lives and work practices, raise efficiency and safety levels, and provide enhanced levels of service. Even more, robotics set to become the driving technology underpinning a whole new generation of autonomous devices and cognitive artifacts that, through their learning capabilities, interact seamlessly with the world around them, and hence provide the missing link between the digital and physical world. Robotics already the key driver of competitiveness and flexibility in large-scale manufacturing industries. So, as you can see, these are the definition of terms that we need to know before we proceed to the discussion. First, automated system composed of elements designed to perform a set of tasks that have been programmed. Chemical synthesis, artificial execution of useful chemical reactions to obtain one or several products. Microreaction, nucleophilic addition of a carbanion or another nucleophile to an alpha-beta unsaturated carbonyl compound containing an electron withdrawing group. Artificial intelligence, or the AI, the simulation of human intelligence in machines that are programmed to think like humans and mimic their actions. And lastly, gas chromatography mass spectrometry, or the GCMS, an analytical method that combines the features of gas chromatography and mass spectrometry to identify different substances within a test sample. So, why we need automated chemical synthesis? Automated chemical synthesis carries great promises of safety, efficiency, and reproducibility for both research and industry laboratories. Most existing approaches are based on automation systems, for example, custom-built fixtures and mechanisms that perform a specific experiment. So in this study, we will go to the introduction of this study. So first, existing apparatus such as mass spectrometers must be modified to be integrated into the automation systems. This prevents those usually expensive pieces of equipment to be used by other systems or by the chemists on other experiments. Second, such systems are not flexible and will require substantial redesign to handle new reactions or procedures. This study aims to make a system based on a robot arm, which by mimicking the motions of human chemists is able to perform complex chemical reactions without any modifications to the existing setup used by humans. As you can see, there are 8 important parts in this experiment. This includes magnetic stirrer, vial, gripper, robot arm, syringe pump, auto-injector, and GCMS. So, magnetic stirrer, a rotating magnet or stationary electromagnet that creates a rotating magnetic field. This device is used to make a stir bar immersed in a liquid quickly spin or stirring or mixing a solution. Vial is a small glass or plastic vessel or bottle, often used to store medication as liquids, powders, or capsules. Gripper is a device which enables the holding of an object to be manipulated. Robot arm consisting of parts linked together in the same way as those of a human arm mounted on a stand. Um, Syringe pump, small infusion device that is used to gradually administer specific amounts of fluids for use in chemical and biomedical research. Auto-injector, a medical device designed to deliver a dose of a particular drug. And lastly, GCMS, an analytical method that combines the features of gas chromatography and mass spectrometry to identify different substances within a test sample. So, in this experiment, the robot can handle liquids, mix chemicals, filter chemicals, can integrate with GCMS, reconfigure, and also to analyze. Godfrey et al. Proposed an automated chemical synthesis laboratory which is capable of modular expansion and allows chemists to remotely guide chemical synthesis for drug discovery. The robot arms were mostly used for pick-and-place operations, but also include diluting of samples, which was briefly stated. So this can be a great foundation for this study. So the researchers do the study because of this recommendation from Godfrey. So in this robot arm, as you can see, modifications such as sensors and tubing were made to most apparatus to facilitate liquid transfer, and all components of the automation unit were dependent on each other. Hence, it will not be convenient to dismantle the unit to access any of these components individually due to the amount of tubing used. This accessibility may be desired by research chemists who share a common pool of equipment, as by isolating equipment in the automation unit, it can result in a smaller amount of resources available. So, as you can see, the researchers develop a system that can help them to execute this kind of experiment. So, a Denso VS60 robotic arm coupled with a robotic hand e-gripper was operated in a structured environment to conduct the experiment. So also, the researchers use an automated and remote syringe pump and fabricated to perform liquid handling, and the GCMS with model Shimadzu GCMS QP2010SE was used for the analysis of the samples created by the robot. In this experiment, microreaction was used in the study. So what is microreaction? So it is the addition of an enolate of a ketone or aldehyde to an alpha-beta unsaturated carbonyl compound at a beta carbon. So the reaction was carried out in a 1.5 ml vial where toluene, cyclopen 2N11 dimethyl malonate were acted using TDB catalyst. The chemicals were distilled and subsequently added to ethyl acetate. The mixture was poured into a cartridge packed with silica powder for filtration, and more ethyl acetate was poured to flush out the analyte sample. So in this slide, we will see the procedure executed by the researchers. So first procedure is liquid handling. Second is mix of chemicals, and then transferring reaction mixture, and again um, mixing of chemicals. After that, filtering, then integrating with the GCMS and reconfiguration, and lastly, the analysis of each sample. So, so, so in this slide, these are the glimpse of the experiment executed by the researchers. So after this slide, I will show you a video where the researchers conduct their experiment about this study. Okay. So in this slide, I will show you a video about the experiment conducted by the researchers. So this is the video. So again, this experiment undergoes a Michael reaction. So these are the procedures in this experiment. So first is grab the syringe pump, then add the cyclopent 2 and 1 to reaction vial, then wash syringe. Then add dimethyl malonate to reaction vial, then wash again the syringe. Add stock catalyst to reaction vial, then wash syringe. Stir chemicals for about 5 minutes. Add reaction mixtures to ethyl acetate, then wash again the syringe. Stir chemicals for about 5 minutes. Pour chemicals into filtration cartridge, wait for filtering. Flush filtration cartridge with ethyl acetate, wait for filtering. Place the final product into the GCMS rack. So there are two runs in these experiments, but again, um, what you did in what you do in the first run, and also again you will do again in the second run. So that's all. Let's go to the next slide. So the analyte samples created by the robot in experiments 1 and 2 were sent for analysis. From the analysis in this table, as you can see, it proved that the robot was capable of conducting chemical synthesis. The average yield for run 1 of both experiments was 30.5%, and the average yield for run 2 was also 30.5%. In addition, the overall mean yield across both experiments was 30.5%. The concentration is directly proportional to the yield and is dependent only on the yield. Hence, the average concentration across all runs was 8.2 mg per ml of analyte sample. So it showed that the robot was capable of achieving consistent yield for both runs of each experiment, even though the experiment 1 and 2 were conducted on separate instances. A new batch of chemicals was used for each experiment. So again, um, automated chemical synthesis using a robot arm and achieve an average yield of 30.5 and average concentration of 8.2 mg per ml of analyte sample form. The experiment proved that robot arms have the ability in automating experiments and are not limited to pick and place tasks, which was seen in previous studies. Taking advantage of the dexterity of the robot, we were able to integrate independent systems together to carry tasks to complete the experiment. So, in conclusion, based on the advantages attributed to robot arms, such as consistency of results and abilities to integrate dependent systems together, robot arms can be used to automate chemical synthesis. So, these are the references used in this presentation. So again, thank you for listening and have a nice day and stay safe always. Thank you.